Continuation of the previous video. Structural variations in constant region domain. That is, biological effector functions are performed by immunoglobulin are determined by the amino acid sequence of constant domain. The CH1 and CH2 domains hold together the VL and VH domains of immunoglobulin via disulfide bonds and extend the Fab segment by virtue of the hinge region, thereby facilitating the conformational rotation of two fragments and thus contributing to epitope binding also. Thereby facilitating the conformational rotation of two fragments and thus contributing to epitope binding also. So CH1 and Cl domain allow random association with VH and VL domains, thereby leading to a diverse repertoire of antibody molecules, antibody diversity. Thus random gene rearrangements of immunoglobulin generate antibody diversity contributed by the constant region domain. Other constant region domain, the immunoglobulin IgM and IgG comprised of 4 constant domains of heavy chain without any hinge region, while IgA, IgG and IgD contains 3 constant regions with CH1, CH2 joined by the hinge region. CH4 and CH3 of 2 identical heavy and light chains contribute as the carboxyl terminal ends of IgE and IgM and IgA and IgG and IgD respectively. The solubility of immunoglobulin in serum is virtue of the oligosaccharide side chains present at the heavy chain of constant domain CH2 of IgA, IgG, IgD, while CH3 domain of IgM and IgE. These CH3 domains of immunoglobulin IgM and IgE are highly accessible for the stimulation of complement system, thereby contributing to the biological effector functions of the immunoglobulin molecules. Immunoglobulin is classified into 5 classes and further subdivided into subclasses. IgG has 4 subclasses, IgA with 2 subclasses. The subclasses can be either membrane bound MIG or secreted immunoglobulin STG. Both differ in their structure and function as at the carboxyl terminal domain. The membrane bound MIG contains a short cytoplasmic tail, a transmembrane sequence and a 26 amino acid long extracellular sequence at the carboxyl terminal domain. The SIG have a variable length hydrophilic sequence at the carboxyl terminal. The spacer sequence and cytoplasmic tail vary among all isotype of immunoglobulin. Fab fragment, FC fragment. VL, VH, Cl, CH1. Then VH, VL, CH1, Cl, CH2, CH2, CH3, CH3. Variable region, variable heavy chain, variable light chain, Cl, CH1, like that. At different developmental stages of humoral cells or B cells, B lymphocytes, different membrane-bound immunoglobulin are expressed. The membrane-bound IgM is expressed by the immature pre-B cells, while mature B cells co-expressed MIGM and IgD prior to any antigenic encounter. Antigenic stimulation triggers the differentiation of mature B cells into plasma cells and memory cells. Plasma cells express various isotypes of secreted immunoglobulin via isotype switching, while memory cells express membrane-bound immunoglobulin of MIGE, MIGG, MIGM or MIGE. Immunoglobulin as immunogens. Immunoglobulin are proteins which can induce an antibody immune response, thus acting as a potent immunogen. These are potential tools to study humoral immune response and B cell development. 3 major categories of antigenic determinants are classified on the immunoglobulin molecule: isotype, allotype and idiotype determinants. Isotype determinants on the basis of differences in the constant domain of heavy chain, immunoglobulin is categorized into classes and subclasses. Similarly, light chains are categorized into its types and subtypes based on the constant region of amino acid sequence differences. These amino acid differences are called isotypic determinants. A unique and separate constant region encodes each isotype carried by all the members of a species. Different species inherit different isotypes. A reason why antibody of one species is recognized as a foreign molecule by another species. Mouse IgG1, mouse IgM. So on B lymphocytes activation, the heavy chain genes undergo class switching, a phenomena producing different immunoglobulin classes and also termed as CSRs or class switching recombination. The plasma cells produce IgG, IgE or any other class of Ig from IgM via class switching due to change in the constant domains of heavy chain. The variable domain of heavy and light chain does not undergo CSR. Hence, the antigen binding specificities of isotypes antibodies has no changes. The immunoglobulin are classified into 5 major isotypes based on the amino acid sequence differences at the constant region of heavy chain, contributing to the structural and functional properties which are class specific. The 5 isotypes are immunoglobulin A, IgD, IgE, IgE and IgM. Stem cell, pro B cell, pre B cell, IgM, immature B cell, IgM plus IgD, mature B cell, activated B cell, sub isotype switching, IgG, IgE, IgA, IgM. Then immunoglobulin A. Immunoglobulin A comprises of 4 polypeptide chains in its monomer form, but it may exist in dimeric, trimeric or tetrameric forms joined by the J chains and called secretory immunoglobulin as present in the external secretions. The human immunoglobulin A has 2 subclasses IgA1 and IgA2, and the heavy chain of IgA is alpha type. It constitutes 10 to 50% of total immunoglobulin present in the serum, predominating in the secretory fluids like tears, saliva, breast milk, and mucous secretions at different organs. The secretory immunoglobulin comprised of dimer or tetramer form, J chain, and secretory component unit of transporting Ig across cell membranes. The J joining chain polymerized the immunoglobulin in its dimeric form, as in case of pentameric IgM. The monomer form of IgA was 160 kilodalton MW, and the secretory component is of 70 kilodalton molecular weight secreted from the mucous membrane lining epithelial cells. Here you could see 1, 2, 3, 4. A dimer from IgA immunoglobulin. Heavy chain is by blue. Look at this. Then blue or violet. Yeah. Light chain, red colored. Light chain, J chain 3 in the middle. Then secretory protein 4, yellow colored. The Fc region of immunoglobulin binds with one of the 5 immunoglobulin-like domain of secretory component and stabilized by disulfide bonds between the Fc region of dimeric IgA and first Ig-like domain of secretory component. Plasma cells are populated at the mucous membrane surface. Secretory IgA in abundance than any other immunoglobulin. The pentameric poly-Ig receptor is expressed on the glandular epithelia of lacrimal, salivary and mammary glands and on mucosal epithelia, urogenital tract, lungs, digestive tract. Then the plasma cells migrate to submucosa of mucous membrane and secrete immunoglobulin A, which tightly binds with the poly-Ig receptor and stimulate coated pit formation, thereby leading to endocytosis of IgA receptor complex via receptor-mediated cellular uptake pathway. These vesicles carrying IgA receptor complex, when fuses with the luminal plasma membrane, the receptor is enzymatically cleaved from the luminal membrane and released together with IgA molecule as secretory component in the mucous secretion. In the protease-rich microenvironment of mucosal membrane, the polymeric Ig receptor protects the hinge region of dimeric IgA molecule from early proteolytic cleavage. The polymeric receptors also interact with the J chain of IgM molecule and enable IgM transportation into mucous secretion across cell membrane. Diagrammatic illustration on formation of secretory IgA molecule as a transport through epithelial cells mucosal membrane. Plasma cells, dimeric IgA, poly-Ig receptor, submucosa, epithelial cell, vesicles, proteolytic cleavage, secretory IgA into the lumen. Secretory IgA are effective molecules at the mucosal sites of various organs. Mucosal sites are the major sites to encounter most pathogenic agents. Binding of secretory IgA to multiple epitopes of large antigens such as viral or bacterial surface antigens can block the attachment and colonization of infectious agents on the mucosal sites. Secretory IgM provide line of defense against several viruses and bacteria such as influenza, polio, reovirus and Vibrio cholerae, salmonella, Neisseria gonorrhoeae respectively. An infant's immune system is not fully developed during the first months. Therefore, secretory IgA molecules through breastfeeding can maintain the health of a baby by protecting it against infections. Then, immunoglobulin D. Immunoglobulin D is expressed on mature B cell surface and like IgM, It is a major membrane-bound antibody molecule with no biological effective functions. IgD helps in stimulating B cells by antigens. So, IgD exists in monomer form with delta as heavy chain component and 150 kilodalton MW. It constitutes 0.2% of total serum immunoglobulin and 0.03% 03 mg per ml serum concentration. In humans, there is no subclass of IgD molecule. IgD probably function in activating or suppressing lymphocytes. They cannot cross placenta, active complement system, or cause mast cell granulation release. Monomer IgD, IgE, IgG. Dimer IgA, pentamer IgM. General structure of immunoglobulin molecules IgD, IgE, and IgE. DAM. Immunoglobulin E. Bransitz and Kussner in 1921 suggested allergic reactions are a response to serum component presence. When a non-allergic person is injured dermally with the serum of allergic person and again injected at the same site, develop a flare reaction called PK reaction due to IgE activity. IgE antibody mediated hypersensitive reactions develop asthma, anaphylactic shock, high fever, and high symptoms. Although the serum concentration is 0.3 mg per ml for IgE, it is a potential and critical biological effector molecule existing in its monomer form. So, binding of receptor bound IgE molecule to allergen and with its Fc region to the membrane receptors on mast cells, natural killer cells, and basophils leads to antibody dependent cell mediated cytotoxicity by the release of granules from mast cells and basophils releasing pharmacologically active mediators leading to allergic manifestations. IgE molecules also provide anti-parasitic defense systems. Let's see that monomeric form of IgE. Then immunoglobulin G. About 80% of total serum Ig consists of IgG, which is considered as most abundant class of Ig in serum. The molecular weight is 150 kilodalton and structure comprised of two identical heavy chains of gamma type and two light chains lambda and kappa. Immunoglobulin class IgG categorized into four subclasses IgG1, IgG2, IgG3 and IgG4, numbered and differentiated in their serum concentration and gamma chain sequences. The four subclasses of immunoglobulin G are structurally differentiated on the basis of hinge region size, position and number of disulfide bonds between the heavy chains, and thus effects of biological functions between subclasses such as IgG1, G2, G4 can cross the placenta and critically important for neonatal immunity development in fetus. Immunoglobulin acts against pathogen and their toxins besides stimulating complementary system of the body. The IgG forms complex with the microorganisms and activate the complement system. IgG3 is the most potent activator followed by IgG1 and IgG2 as a complement activator. IgG1 and IgG3 are excellent opsonants which enhance the phagocytosis of other innate immune cells such as natural killer cells, macrophages, etc. by binding with Fc receptors on their surface. IgG4 and IgG2 have less affinity towards Fc region receptors, therefore does not contribute much in opsonization. IgG1 hinge region, heavy chain, IgG2. General structure of IgG immunoglobulin molecule subclass IgG1 2 bonds and IgG2 4 bonds which differ in number and arrangement of disulfide bonds. IgG1 1 bond, IgG2 4 bonds. Light chain IgG3, IgG4. General structure of IgG immunoglobulin molecule subclass IgG3 11 bonds and IgG4 4 bonds which differ in number and arrangement of disulfide bonds. Then immunoglobulin M, the total serum concentration of IgM is 1.5 mg per ml and constitute 10% of total serum immunoglobulin. IgM is a membrane-bound monomeric immunoglobulin on the surface of B lymphocytes on antigen binding B cells differentiate to plasma cells secreting pentameric form of IgM of molecular weight 900 kilodalton. Monomeric MW is 180 kilodalton. The pentameric form of IgM is held together by their carboxyl terminal Fc region, more specifically C-mu4, C-mu4 and C-mu3 domains by virtue of disulfide bonds. This combined structure of 5 monomeric forms differ 10 antigen binding sites facing towards periphery of the pentamer molecule while effector Fc region at the center. Besides bisulfide bond between heavy chains of monomers, a polypeptide chain called J joining chain is linked to two monomers of IgG with the help of its disulfide bonds. This is IgM, representing the pentameric form containing a J chain. The additional Fc linked J chain binds to the cysteine residues of the carboxyl terminal ends of 2 of the 10 mu chains. Just before pentamer formation of IgA molecule, a J chain polymerizes the monomer forms. There is no subclass of immunoglobulin M. And on antigenic stimulation of B lymphocytes in primary immune response, first immunoglobulin class produced is IgM. It is the first immunoglobulin molecule produced by newborn. Serum IgM contribute to maximum valency of antigen binding than any other isotype. Although IgM molecules can bind 10 molecules of hapten, but in case of longer antigens and due to steric hindrance, only B5 can bind simultaneously. The pentamer form of IgM is potentially efficient than other isotypes in virtue of its multivalent activity against multidimensional antigens. Briefly considered, RBCs are multidimensional antigens. Its incubation with antibodies leads to agglutination. In comparison to IgM, any other class isotype will require 100 times more molecules to achieve the same agglutination status. IgM is most efficient in complement system activation. A pentameric IgM molecule is non-diffusible because of its large size. Like IgA molecule, it also forms complex with secretory components which allow IgM transportation through epithelial lining to mucosal surface and thus act as an important secretory immunoglobulin. Then allotypic determinants. Although isotypic determinants inherit in all members of a species, some genes exist multiple allelic forms encoding amino acids differences in some members of a species called allotypic determinants or allotropes. Allotropes has been characterized for all 4 IgG subclass of gamma chain, 1 IgA or alpha chain and 1 kappa chain. The gamma heavy chain markers are referred as GM markers. Depending on the allelic number, class and subclass type, total 25 GM markers are identified. For instance, G4M, G1M, G3M, etc. Among IgA subclass, only IgA2 has allotropes. For example, A2M and A2M2. These allotropes exist for kappa chain and Km. Allotropic determinants usually differ to 1 or 4 amino acid residues. Mouse IgG1 strain A, allotypic determinants, mouse IgG1 strain B. Idiotypic determinants. The amino acid sequence at the variable domain of heavy and light chain play dual function as function 1 as an antigen binding site and via conformational changes in variable regions as antigenic determinants, also referred as idiotype or idiotypic determinants. In some individuals, the idiotypic determinants are the sequence of variable region outside antigen binding site, while an idiotype may be the actual antigen binding site also. Idiotype, mouse IgG1 against epitope A, mouse IgG1 against epitope B. Then B cells of same clone produces antibodies are recognized by identical variable regions and thus have some idiotype. Multiple idiotypes are present on individual antibody molecules and the sum total of all idiotypes referred as idiotype of antibody. Summary: immunoglobulin is glycoprotein molecule with 4 polypeptide Y-shaped structure. The divalent Y-shaped immunoglobulin structure is related to their structural and functional variations, antigen binding specificity, diversity, and genetic control. Any foreign molecule can elicit an immune response in the host body, and the response is specific and diverse. This diversity is attributed to the recognition and binding of diverse antigenic determinants or epitopes to antibody molecules with different affinity and specificity. Immunoglobulin repertoire is a general term used for the sum of all antibodies with different specificities. The immunoglobulin is a Y-shaped polypeptide structure comprising of 2 identical heavy chains and 2 similar light chains. Each heavy chain has 1 variable domain and 3 to 4 constant domains. Each light chain has 1 variable domain and 1 constant domain. These polypeptides are interconnected and stabilized by the disulfide bonds. Some immunoglobulin have hinge region connecting variable domain and constant domain of heavy chain, also contributing to the flexibility of the immunoglobulin which aid in effective functioning. Among different means factors creating broad range of immunoglobulin repertoire, somatic hypermutation in the genetic segments of the variable domain act as a critical factor in determining the extraordinary diversity of immunoglobulin. Besides SHM, which means point changes in the genetic material of rapidly proliferating activated B cells, different gene rearrangements and combinations of hypervariable regions or CDRs, complementary determining region or heavy chain variable region, also called a class switching, also contribute to diversity of immunoglobulin. Each isotype has separate expression of constant domain genes, hence immunoglobulin isotypes are dependent on the constant region of heavy chain. Based on the constant region domain differences, immunoglobulin are classified into 5 isotype: IgA, IgD, IgE, IgG and IgM, all differing in their immunological functions and under different conditions and different timing respond differently to some epitopes or antigenic determinants.